Hey guys, I'm Richa and welcome to the iBiologist. In this video, we will discuss the cell organelle Golgi body. Golgi body term has been given from the scientist named Camillo Golgi, who had been given its morphological structure while he observed in nerve cells of cat and barn owl. While it is firstly observed by the George, its ultrastructure was given by the Dalton, though it's called the Dalton complex. It's also known as lipochondria. In plant cells, it's also called the dictyosome. Its size is varies according to its function. It is bigger in secretory cells while smaller in non-secretory cells. As liver and pancreatic cells are secretory cells, while muscle cells are non-secretory. In vertebrate cells, number of Golgi is 1. But in plant cells it varies up to 6 to 12, while lower algae has 1. In rhizodial cells of Cara has few hundred of Golgi complex. Let's talk about its position. In vertebrate cells it is perinuclear, it means it's closer to nucleus, while in plant cells it is diffuse or scattered. Clear cytoplasmic area around Golgi body is called zone of exclusion. It is pleomorphic organelle, which means its shapes change according to its function. Such organelles called pleomorphic organelles. Now we will discuss its structure according to Dalton. According to Dalton, Golgi is categorized in three elements. There is strike-like structure known as cisternae, the large spherical structure known as vacuoles, the small spherical structure which is known as vesicles. Golgi is polar in nature. The cisternae is slightly curved and its ends are swollen. They are placed on one or another in stag-like structure, and the cisternae may interconnected with the help of small connections. Vacuoles are normally in 600 angstrom in diameter, and it's normally found closer to concave side. Because of this arrangement, we have seen there is concave side and convex side. Concave side is closer to the nuclear membrane, while convex side is outer side towards the plasma membrane. Concave side is known as formative phase or cis phase, while convex side is known as maturation phase or trans phase. From the nuclear membrane itself arises the endoplasmic reticulum. From the endoplasmic reticulum, the vesicles which have come, either they have protein if they come from rough endoplasmic reticulum, or either they have lipid if they come from smooth endoplasmic reticulum. So these vesicles which are coming from the ER, they will come near the Golgi body and release the substance they have into the Golgi body. That means the raw material coming from the formative phase. The substance through the connection goes to the swollen part of the Golgian vacuoles. And the Golgian vacuoles is going to become bigger and bigger and it gets pinched off. And this is going to become the vacuoles, which may carry the hydrolytic enzyme which we call lysosome. That means from outer part or convex part of the Golgi, the raw material transform into the final product, and that is why this side is known as maturation phase or trans phase because of the transformation, and the concave side the process of formation is gonna start. So this side is known as formative. The fungi has only one cisternae. That is why it is called the unicisternal. The Golgi apparatus is responsible for transporting, modifying, and packaging the material into the vesicles for delivery to targeted destinations. So basically, Golgi is used as packaging bag for cytoplasmic material. I have created a separate video which covers the Golgi functions in detail. You can find its link in the description box below. Before the video ends, let's recall the Golgi parts along with ER. Here you can see the green part which is nuclear membrane and it has the pore-like structure which we call nucleus pore. Along with nucleus, there are two types of endoplasmic reticulum is there. For differentiate, the smooth ER and the rough ER is its protein. Smooth ER has lack of protein on its outer membrane, thus it is smooth, while rough ER has the protein on its wall. As we discussed earlier, concave side or maturation phase is towards the nucleus membrane, which is numbered as 9 here. You can see the stacks of cisternae create the Golgi here. And the trans phase or formative phase is numbered as 10. In this figure number 6, you can see the raw material coming into the Golgi. It is first entered into the vesicles and then the vacuoles.